This is St. Louis on the Air from St. Louis Public Radio. I'm Elaine Cha. The Diamond Laboratory in St. Louis is not where you'd go to shop for a wedding ring. Staffed by dozens of scientists at Washington University, this lab tries to understand viruses. And for the last several years, it has been studying COVID-19. Dr. Michael Diamond heads the Diamond Laboratory. His work on understanding the building blocks of viruses and immunity spans decades. His team created the world's first COVID-19 vaccine delivered through a nasal spray, and they're still discovering new things about how our immune systems react to the COVID vaccine and what the vaccines of the future may look like. In a new study published in Nature, Dr. Diamond and his co-authors took up a future-oriented question. What happens to the strength of our vaccines as we stack booster on top of booster? This isn't a hypothetical problem, and it makes fighting the flu, for example, very complicated. It introduces the possibility that those vaccine boosters at some point might not be quite so boosting. And when we're talking about a global pandemic, that's a scary thought. As it turns out, the researchers found something quite interesting about the boosters for the vaccines for COVID-19. And here to talk about those findings and what scientists are still discovering about the virus and our body's reaction to it, I'm joined by Dr. Michael Diamond. Dr. Diamond, thank you for being on our show.
Thank you for having me.
So before we get to your new research, tell us, what is the Diamond Lab?
So our laboratory studies viruses and how they cause disease. We're very much interested in emerging viruses, and we're interested in understanding what are the steps they take to get into cells, what happens when the immune system tries to combat them, how do they evade the immune system, and ultimately, what kinds of immune responses are critical for controlling these viruses so that they can cause less disease in humans and also in other animals.
Mm-hmm. Now, you study the molecular basis of viruses, Doctor. What is that, and why does that interest you?
Well, viruses are not static, especially the viruses that we study. We study RNA viruses, and so they accumulate mutations in time. And so understanding how these changes occur and what the significance of these changes are, not only for being able to cause more disease in certain cells or certain animals, but also to evade responses that the immune system is trying to generate to protect. This is critical. So in order to do that, you really need to understand on a molecular level what's happening to the individual proteins or individual genes and see over time what is the trajectory of that in the context of changing the landscape of infection and disease.
Mm-hmm. Now, you are the principal investigator for this lab. What does that mean? And if the lab were a sports team, for example, would that make you the coach?
Well, I have many hats. I probably would be the coach, the general manager, if you will. There's a responsibility in terms of interacting on a day-to-day basis with the people in my laboratory. We have many trainees, so we have students that are either graduate students, postdoctoral fellows, medical doctors. We also have visiting scientists. We have staff scientists. We have technicians. And then we have a host of administrative people that we interface with. So those are I interact with every day to see how they're doing on their projects, to provide direction, to also provide critical advice and critically evaluate the projects as well. So it really ranges from day-to-day operation in the science to larger pictures, to securing funding, to working with our federal government partners, if you will. So there's a lot of responsibility as a principal investigator, but really the work is being done on a day-to-day basis by the scientists in the laboratory. Some of them are trainees and some of them are permanent assistants.
So it certainly sounds like there's a lot going on there on any given day. I'd like for you to take us back then to 2020. You were already studying viruses, of course. How is it that the emergence of COVID-19, how did that impact your lab?
Yeah, so we had been through a few of these before. Since I work on emerging viruses, we had started the laboratory really on West Nile virus when it had first emerged in the United States at about 2000. We began studying the original SARS-CoV in 2003. we moved to some of the other emerging viruses like Zika virus and chikungunya virus somewhere in the 2015 or 2010 time period. And so we've become accustomed to using our skills in molecular virology to study new viruses as they emerge or reemerge, if you will.
Mm-hmm.
And we pivot. We basically decide that it is an important virus. There's not a lot known about it. We need to develop models to be able to test therapeutics or vaccines, or even just to fundamentally learn about the basic biology of the virus. And we may switch projects. We may have people volunteer to say, hey, I really am interested in working on this new virus. Or we do this as a team together and divide up responsibilities as we did during COVID. So we collectively we make the decision to work on it, and then and not only in my laboratory but also with other laboratories here at Washington University
Mm-hmm.
and elsewhere.
So, how much of a shift in focus then was there to COVID-19? I mean, if you were already studying other viruses, like how did that all work?
Yeah, so essentially, as you may remember, there was a very difficult situation early in the pandemic where we just didn't have any drugs or any vaccines, and there was a lot of uncertainty and a lot of danger at some level of contracting it and getting sick. And so the university essentially shut down operations except for those that were working on COVID.
Mm-hmm.
And so essentially my lab completely converted to a COVID lab. Everybody who was here was working on COVID, and if you weren't working on COVID, you weren't here. And so there was both an impetus to have people continue to work, but also there was an interest in the project, and also there was a necessity, given that we actually had the biocontainment facilities in the laboratory, to actually study a virus that was potentially dangerous and still is dangerous. And so you need high containment facilities. And because my laboratory worked on emerging diseases and had these facilities, we were poised to make this pivot and study it.
Mm-hmm. Now, to what extent then were you ready for that? And in what ways were you not so prepared?
Well, I don't think you're ever ready for what happened. I think, we expected this to be perhaps a small problem, or like the original SARS was. I don't think we really envisioned the extent that this could happen. Of course, we knew that new viruses, which the population has no pre-existing immunity, could be very dangerous. But what we didn't anticipate was the way that it spread, and the fact that it spread in people who were not symptomatic. I think that was really a surprise, and that really changed the ball game in terms of the numbers of people that got infected, the numbers of people that got sick, and also the severity and urgency of the problem. And so we were prepared at some level of having some of the facilities. We even had expertise in my laboratory in coronaviruses, even though I'm not per se, was not at the time a coronavirus expert.
Mm-hmm.
We had postdoctoral fellows and others who had worked on coronaviruses in their prior training. So we were prepared at that level, but really not for the extent of the disease and what it would take to actually study something from scratch, never having seen it before.
Right. What was the kind of improvisation that was involved in getting things together? And were there any unexpected obstacles? I mean, yes, COVID-19 was something that nobody had ever seen before, but were there other things that stood in the way, hurdles that you had to clear that you had not anticipated?
Well, I think there was several things. First of all, the institution was great. They supported us prior to the point that we actually got any federal money. The dean and the medical school and the chancellor all garnered resources, not just for my group, for other groups. Then there was tremendous collaboration and interaction with members of other departments, my department in medicine, other departments, the chair of microbiology, Sean Whelan, and others, to be able to facilitate interactions with their scientists such that we could collaborate and use collectively the space that was available and the resources that were available to do particular experiments.
Mm-hmm.
I think, what the challenge was in some ways was even just generating the reagents, getting the virus. We had to lobby the CDC to get access to the virus. We had to make sure we had appropriate institutional biosafety committee write-off. We had to make sure that we were protecting people. There was certainly in the population concern about studying a virus to some degree, which we didn't know that much about. If you remember, there was a lot of controversy, still is, about what occurred in China.
Right.
So studying it really created a number of things that we had to have in place for biosecurity, for biosafety. All of those in my mind, I knew existed, but of course, at the time when they were occurring, really needed to make sure that we had all those in place to make sure that your people are safe while you're trying to get answers to fundamental problems of significant importance.
Mm-hmm. Dr. Diamond, what were the most pressing tasks at the beginning of the pandemic, and what questions were you trying to answer?
Yeah, I think there was really three things that we were felt that we were in a good place, meaning that we had the right people, the right expertise, and the right history to study. One of them was to generate an animal model so that we could actually look at countermeasures, so that we could very quickly figure out whether something worked or didn't work. And so we didn't know at the time whether this virus would infect conventional laboratory mice, and it did not, in fact. And so we had to rapidly figure out how to get this virus to work in mice or in other animals. That was the first thing. The second thing was to develop a vaccine. We felt that we had expertise previously in doing this, and we were very much interested in developing what are called mucosal vaccines that might be delivered intranasally or through mists or otherwise that could actually stop the virus from being transmitted. We knew that there were individual laboratories and companies that were pursuing what I'll call systemic vaccines, ones that are delivered intramuscularly. Mm-hmm. But we knew that those could work potentially, although of course they were more successful than we ever imagined. But we also felt that it would be important to have different approaches to vaccinology against this particular virus. And then the third thing we worked on was antibody-based therapies. These would be a treatment that could be used immediately. And so we collaborated with a group at Vanderbilt University to generate and evaluate monoclonal antibodies that ultimately became part of the drug Evusheld, which was used for a long period of time.
Mm-hmm. I'd like to hear more now about the new study on COVID-19 vaccines, and we mentioned a little bit about that in our introduction. Now, there's something called imprinting. Now, the takeaway through all of this seems to be that there are not only boosters that are not getting weaker, there's indication that they're getting stronger, even to variants that we've never seen before. And it has to do with our immune system's ability to remember, or maybe quote, remember things. And that is that imprinting. Dr. Diamond, help us to understand the study. What is imprinting, and what does it have to do with our COVID boosters?
Yeah, so the concept of imprinting is essentially how immunologic memory works. And so the first vaccine that somebody receives induces an immune response. And part of that immune response is you have cells. They're called B cells. These are the ones that ultimately generate antibody. But these B cells also stay around for long periods of time in different places, usually in lymphoid organs like lymph nodes and other places. Mm-hmm. And they're sitting there, and there's a subset of these B cells which are just sort of sitting and waiting. But they've already been primed to recognize a particular vaccine or a particular protein of a virus, if you will. And then a second time you may get vaccinated, those cells can immunologically remember the first protein that they saw, and then they can respond very rapidly. That's memory. And the reason why we have that is so that if we get infected a second time, we're much more poised to respond quickly so that we can rapidly control an infection. And that's the whole premise of it. In the context of the vaccine, we were interested in understanding now what happens when you see something that's not exactly the same as the original one that you saw. So this is what is called a variant vaccine, or a variant antigen or variant protein. And how does that affect the response? There are two possibilities. One of them is that you get a completely new response against the variant protein, and the other is your response is being shaped by the original memory you had against the first protein. And because of that, you're focused now on shared parts of that protein. So one that was in the original protein and one that's in the variant protein, and now the response gets directed against that conserved shared region as opposed to any new region of the second protein. Mm-hmm. So this is the concept of imprinting. So imprinting is how memory works. And imprinting can be positive, negative, or neutral. And you sort of alluded to it in the beginning segment, part of the segment, where in flu, we've seen flu many, many times. And so what happens is now our memory focuses on certain regions when we get boosted. But unfortunately, those regions that we're now making these boosted responses to are not helping us control the virus. So that's considered a negative outcome of imprinting.
Mm-hmm.
So what we were surprised at, I guess, is there was a goal of making variant vaccines to try to generate new responses. But instead, we basically completely generated responses that were shaped by this first immunologic protein, if you will, the first vaccine, but it was actually positive and not negative.
Mm-hmm. Well, that's a good place to pick up the conversation after we take a very quick break. We will be back shortly to continue this conversation with Dr. Michael Diamond. This is St. Louis on the Air on St. Louis Public Radio. Welcome back. Let's return to our conversation about the development of COVID-19 vaccines, past, current, and future. Our guest today is Washington University professor and viral immunologist, Dr. Michael Diamond. As head of the Diamond Laboratory at Washington University, Dr. Diamond leads a team of more than 30 scientists in research into the molecular basis of viruses and immunity. The lab has been at the forefront of COVID-19 development. Dr. Diamond, before the break, we were talking about imprinting. I'd like to explore a little bit more about the findings. And what you did find is something that seems to a layperson, in any case, really remarkable. And the study found that imprinting was actually having a positive effect, which you had mentioned, as it drives expansion of cross-neutralizing antibodies. That's a quote from that study. Now, those antibodies are a big deal because they inhibit emerging variants and distantly related sarbecoviruses. So, Dr. Diamond, those distantly related viruses and emerging variants, it sounds like you're saying our bodies can protect itself against those too?
Yeah, that was, I guess, the most surprising thing. We expected that we would get protection against the variant. We were thinking that we might get protection that was mediated by specific antibodies. But in fact, as I said, they were shaped by the original one. So they're all cross-reactive. And now they cross-react to the variant that was in the vaccine. But they also cross-neutralized other variants, ones that were even came after the vaccine, that were even newer variants, if you will. Mm-hmm. And then what was really surprising was that these variants had driven the antibody response to such a cross-reactive state that they could actually neutralize viruses that are not yet in circulation, that are what we call pre-emergent viruses in the family. So these are highly conserved epitopes. These are regions on the viral spike protein, which are present in many of these different related viruses. And now these cross-reactive antibodies that are driven by the boost that you received are actually able to inhibit not only the variants exist now, that existed at the time of the vaccine, ones that exist now, and then ones that we anticipate will exist in the future, and even ones that might come with a new pandemic virus if it ever occurred.
Mm-hmm. So what are the implications then of this study? I mean, does it change the big picture about boosters, for COVID-19, getting stronger, iteration after iteration?
Well, it's not so much that they're getting stronger. I think that the magnitude of the response is not actually perhaps as strong as just when you've got the vaccine the first time, because the cross-reactive responses are different than what we call type-specific or focused just on one virus. What we're driving is this breadth of response, and one key question that we don't know is how long do these last? And so early in the pandemic, we saw rapid increases and rapid decreases, in part necessitated the frequency of the boosters. Over time, what we've seen is a more durable response. What we don't know is, are these cross-reactive, cross-neutralizing antibodies, how durable are they? We looked at a relatively early time point, these people are still being followed who were the ones that were sampled. And I think we need to know over time, how long do these cross-neutralizing antibodies last? This will help us predict perhaps how often we need to be boosted.
Mm-hmm. The future of vaccines is obviously something that you're thinking about. And in 2022, you'd created a COVID vaccine that is applied through a nasal spray. You mentioned the mucosal. That was something that was approved, that is, in India, but it was not approved in the US. What is it that is different about a nasal spray vaccine compared to an injected form?
So an injected vaccine generates robust both B-cell and T-cell responses that are circulating through blood. And they can go to many places in the body, but it takes them time to get there. A mucosal vaccine generates immunity at the site of where it's introduced. So, for example, a nasal vaccine would introduce immunity in the nasal passageway, also in the upper respiratory tract, in addition to generating systemic responses. So in theory, you would be able to prevent infection at the portal of entry and potentially transmission. That's at least the theory, whereas the systemic vaccines protect against disease, but they're not as good as protecting against infection or transmission. And so that's one of the reasons why we still see constant infections. It's not that the vaccines are not working. Mm-hmm. They cannot protect against infection per se, meaning some low-grade fever, sore throat, nasal congestion, or cough. They're protecting you against severe lung disease, hospitalizations, and death. They're very good at doing that, and they're still good at doing it. They're just not good at protecting against infection, whereas a mucosal vaccine might have that advantage.
Mm-hmm. Well, we wish you the best of luck as you continue in the work that you are doing, and perhaps we'll check back with you about what other research you've been working on and what developments have come to light. Dr. Michael Diamond is a Washington University professor and viral immunologist. Thank you so much for talking with us today, Dr. Diamond.
Thank you for the opportunity.
This episode was produced by Danny Wissentowski.
Audio engineering and podcast design by Aaron Dorr.
Our executive producer is Alex Hoyer.
St. Louis on the Air is a production of St. Louis Public Radio. Understanding starts here.
Do you find yourself regularly listening to episodes of St. Louis on the Air? Suggest us to a friend you think might enjoy our conversations, and leave us a review and rating on Apple Podcasts on the App Store. It's the simplest way to help people discover our show. Thank you.
St. Louis Public Radio is a member-supported service of the University of Missouri St. Louis.
The Gateway brings you the day's news from the St. Louis region and across Missouri. It also includes stories from the Illinois side of the river and our Metro East reporter, Will Bauer.
In Alton, in Belleville, in East St. Louis, in Edwardsville, in Cahokia Heights, at Scott Air Force Base, I'm Will Bauer. St. Louis Public Radio.
Listen to reports from Will and all of our journalists weekdays on The Gateway, on the STLPR app, and wherever you get podcasts.